Good evening, everyone. Welcome to BioVanguard Center presentation. For today's session, Yash Karnani is our presenter, and he will be discussing on the topic Deep Learning Integrates Histopathology and Proteogenomics at Pan-Cancer level. So before the presentation, I would like to introduce Yash. Yash Karnani is currently working as a junior research intern at Renix Life Sciences, exploring clinical applications of wearable devices and extracting information from medicine strip for potential use in health monitoring and medical diagnostics. He is pursuing BTech in computer science and IoT at Geetham University, and his academic background includes artificial intelligence, IoT, and data science with projects in computer vision and web development, as well as an internship as a Java developer at Woodpecker Analytics. His experience at Renix has helped him to strengthen his expertise in artificial intelligence and data analytics, which enables him to contribute to innovative healthcare solutions. He aspires to leverage technology to drive impactful advancements in data driven decision making. Now, I welcome Yash to start with your presentation.
Thank you, Ma'am. Good evening, everyone. I am Yash Karnani, and today I'll be presenting a fascinating study titled Deep Learning Integrates Histopathology and Proteogenomics at Pan-Cancer Level, published by Wang et al. In Cell Reports Medicine. This research pushes the frontier of AI in cancer diagnostics by uniting pathology, transcriptomics, and proteomics. Histopathology remains a mainstay of cancer diagnostics. However, it has limited ability to infer molecular characteristics. On the other hand, proteogenomics provides deep insights but is expensive, time consuming, and not scalable. This study aims to bridge these modalities using deep learning to make molecular insights accessible from standard H&E slides. The core objective was to develop a deep learning model that could predict not only just histological but also clinical and molecular features from pathology slides. More importantly, the model is designed with interpretability in mind to aid human pathologists, not to replace them. Data sources. The training data comes from CPTAC with 657 patients and over 2700 H&E stain slides across 6 cancer types. Validation was done on 5374 slides from TCGA. What's impressive is that transcriptomic and proteomic data were paired with these images, allowing multimodal training and correlation. What makes this study unique? So there are three main key innovations. First, it's the first large-scale study to integrate proteomics, not just transcriptomics, with histology through deep learning. It proposes a task classification scheme, class 1 versus class 2, based on human interpretability. It enhances clinical interpretability through expert pathologist feedback, setting a new standard for explainable AI in medicine. The Panoptes architecture. The authors designed a multi-resolution CNN called Panoptes, based on Inception ResNet. It ingests image tiles at 2.5x, 5x, and 10x magnification, just like how a pathologist would inspect a slide at different zoom levels. The model was trained with a weighted softmax loss and optimized using Adam. Here is how you see a schematic of Pantelop's model. It mimics how a pathologist works by looking at low, medium, and high magnification views of the same slide. The three resolution input is shown here: 2.5x, 5x, and 10x tiles combined using a CNN backbone. The classification scheme is crucial for understanding the study. Class 1, tasks that pathologists already perform, like tumor or normal. Class 2A, AI discovers pattern and humans can learn to recognize them after review. Class 2B, patterns not intuitively understood by pathologists only discoverable through AI. For example, predicting tissue organ origin is class 1, TP53 mutation is 2A, and staging is 2B. The task performance. Let's talk results. The model achieved exceptional AUROCs. The tumor versus normal AUROC is about 0.0995 and the tissue of origin is 0.979. Even class 2 tasks like TP53 mutation were predicted with high AUROC of 0.86. These models generalized well to TCGA data, especially for formalin fixed slides. On this slide, the heat map and AUROC scores demonstrates how the model classifies different tissue origins and tumor versus normal states. The TSNE plot shows how well the learned features separate by tissue types. Each cluster represents a unique cancer subtype. On the right side, the saline maps on the saline maps show which parts of the slide the model focuses on when predicting TP53 mutation. Notice the emphasis on nuclear atypia and inflammation, which aligns with the known pathology of TP53 altered tumors. Human AI collaboration. This is where things get exciting. The authors didn't just stop at prediction, they involved pathologists in blind reviews of model outputs. They found that AI uncovered morphological clues like tumor invasiveness and immune cell density that humans could just recognize, suggesting a true partnership between human and machine. Proteogenomic integration. To connect image features with biological mechanism, they used sparse multi-CCA to correlate imaging data with transcriptomic and proteomic features. This revealed pathway level signatures such as IL-1 signaling in tumors with specific mutations like TB53. The canonical variate plots here show how image features correlate with transcriptomic and proteomic signals. Each variate captures a specific biological process, like cell cycle regulation, which is visually reflected in the mitotic morphology. TP53. Here's a detailed look at TP53 mutation prediction. The mutated samples show increased inflammation, mitotic activity, and immune infiltration. Features confirm at both proteomic and histological levels. Interestingly, the model captures monster cells with huge multilobulated nuclei. In TP53 mutant tumors, something pathologists later validated. These images show the histological differences the model uses to distinguish TP53 mutants, particularly increased inflammation, disrupted architecture, and giant pleomorphic nuclei. These features align well with the proteogenomic findings on immune cell infiltration. Clinical implications. These models can support early cancer screening, fast intraoperative decisions, and cancer typing in resource limited settings. This is a step towards scalable and explainable AI in cancer care. The Panoptes web interface, a user-friendly web app, was developed by clinicians to test the model on their own H&E slides. It shows slide-level predictions, tile-level heat maps, and prediction probabilities. You can access it on their website. A few limitations include lack of spatial heterogeneity modeling. Adjacent benign tissue might makes biased predictions. Batch effects in public dataset. Still, the model showed robust performance and generalizability. To summarize, deep learning can predict molecular signatures from H&E slides. Integration with proteogenomics adds depth and biological relevance. The human AI partnership improves both performance and interpretability. This work sets the stage for future clinical AI deployment. Thank you for your time. Feel free to connect with me on LinkedIn.
Yeah. Thank you, Yash, for the presentation. Audience, now the floor is open for the queries. If you have any questions, you can type in the chat box. Okay, I don't see any questions. So with this, I think we can conclude the session. Thank you, Yash, for the presentation, and I thank each and everyone for attending the session and hope to see you all in the next presentation.
Thank you.
Thank you.